Hello and welcome to the Legends of Biotech series on Biotecnika's YouTube channel. I'm your host Shekhar, and today is the episode number 1. And today we have a special guest with us, and he is going to share his journey and a lot of challenges which he faced and how he overcame. And he's also going to leave a indelible mark on your minds because his work in the biotech industry is huge. So, ladies and gentlemen, boys and girls, today I have Dr. Subramanyam Vangala, who is a seasoned biopharmaceutical scientist and executive with 30 plus years of experience in drug discovery and development. He has held various leadership roles in global pharmaceutical companies in USA, including Vet, Johnson & Johnson, Purdue Pharma, and Shire, as well as in Indian contract research organizations such as Sai Life Sciences and Advinus Therapeutics. Currently, he serves as the co-founder and CEO of Reagene Biosciences Private Limited, which is based out of Bangalore, and Reagene Innovations Private Limited, which is based out of Hyderabad. Dr. Vangala's expertise encompasses various facets of drug development, such as drug metabolism, pharmacokinetics, bioanalytical research, clinical pharmacology, and toxicology. He has over 30 plus investigational new drug submissions and has been involved in development of marketed drugs such as Zaloplon, Tigasil, and Canogliflozin. In recent years, Dr. Vangala has focused on innovative approaches in drug discovery, notably the development of 3D printed organs and human tissue models as alternatives to animal testing. Under his leadership, Reagene Innovations created the first fully integrated 3D bioprinted human-like model for type 2 diabetes, which earned recognition at the 5th Annual Conference of the Society for Alternatives to Animal Experimentation in 2022. So let's welcome Dr. Vangala. Namaskar, sir. Welcome to the show. How are you?
Thank you, Shekhar. My pleasure to be on this interesting chat with you and see what we can learn from each other in terms of where the biotechnology has been and where it is going in the future.
So I was just looking at your LinkedIn profile, sir, and it's so huge. I realized that I was 3-year-old when you finished your postdoctoral research, and that's like I was just a kid being born and you had already accomplished so many things. So the last 35 years, I'm sure, has been very challenging as a scientist for you. So we, as audience of Biotecnika, would love to learn from your career journey and motivation. So if you could share a few highlights of your career journey so far.
Yeah, it is, destiny, I call it. Many times we are destined to do something in life. And I wanted to do PhD right after my MSc Biochemistry from Andhra University. And I was looking at various universities in India and especially in the Institute of Science, IIT Delhi and many others. But I got a break in getting admission with scholarship in a Canadian university. So I didn't look back and went to do PhD. And at first it was tough to adjust to the culture there, not only culture, but also our education system is different from their education system. So trying to adapt to it was a little bit difficult. So once you adapt to it, I think there's no stopping. So I finished my PhD in a very highly reputed lab, which actually helped me to grow my career further. I went to University of Toronto for a small postdoc position for a year. Then I went to University of Colorado for 2 years as a research associate. Then I was research assistant professor at Berkeley, University of California at Berkeley, where I was also teaching some courses on biochemistry of cancer, biochemistry of diseases and toxicology, etc. So then a choice came and I got an industry offer. Whether I want to stay in academia or industry, it was a tough choice at the time. I decided to choose industry because I don't have to run and get grants to survive. day to day. So I moved to American Cyanamide Company, which is now Pfizer, basically. This was in 1993, right? Then I spent with a gentleman called Cretonelli, and he and I worked at, he was my boss for 17, 18 years in pharma industry. American Cyanamide, starting with American Cyanamide, then to Purdue Pharma, and then J&J. So I learned a lot from him, not only scientific skills, but actually managerial skills. How do you manage science and business? Right? Those are very critical skills that he actually fine-tuned me for understanding of how do you think in business line versus a scientific line. So that's very critical because you need to always look at what is the business angle for this particular science. It may look very appealing, but business-wise, it may not be great it may not be appealing at all. So with which I had very good career span for 18, 20 years in pharma industry. Then I got an opportunity to come back to India, and Sai Life Sciences wanted me to build their infrastructure for DMPK and some toxicology, exploratory toxicology, etc. So I built that infrastructure for them. And after that, Advinas called me on in Bangalore. So I came and helped them update their infrastructures to the current state of the art. So that happened. After that, I realized that I need to do something for what I like. I've been in drug discovery, as I said, for more than 25 to 30 years now. What I recognized was you take a lot of molecules through the IND program into the clinic, but the clinical is very challenging. Many of the things that you see in the preclinical level are not translating into the clinical outcomes.
Correct.
So, sure, animals are not humans. Rat is not a human, dog is not a human, monkey is not a human. So they all have different biochemistry, pharmacology, and everything. So you need to understand better how the drug testing that done in animals translate to the clinic. So the only thing that I actually stumbled upon a project while I was at J&J, we funded a company called Dural, one of the first ones to come up with microfluidic chips to do the 3D models. And it was a very good learning. I mean, I delegated to my team and we got fantastic data in it compared to the standard old methods people are using like single cell cultures, etc. So that was on my mind. How do I build it back? So we came back to here, Bangalore, and I collaborated with Dr. Shekhar, who was also my fellow mate in PhD. And we created this Reagene Biosciences entity, but we decided to do 3D bioprinting instead of microfluidics with chips. So with that, we actually created 5 or 6 models now, 3D models. Those are very, very good ones to understand how the disease happens in the humans relative to animals. So why we can understand better now how to develop drugs, how to discover drugs in vitro 3D models that can be translated better into the clinical trials. In a nutshell, this is what I'm trying to do right now to translate the outcomes to clinic rather than just do standard old-fashioned drug discovery where it's a cookie-cutter approach. You take in molecules into the clinic and then you don't know what the fate of the drug. Okay?
Right. So in our past interactions also through the iBiome platform, you have always shared your ideas on how the 3D printed bio model is different and better than the animal model. If you could throw some light on what inspired you for doing this and how effective this is.
So the problem with the animals, there are two different things. Animals' biochemistry and pharmacology is different from human pharmacology and biochemistry. Having said that, I mean, animal models many times can be used to get some information how the drug is working or how the drug can be toxic. But the problem comes in when you actually go into the clinical trial, is that relevant or not? So you, based on the animal data, you go into the clinic. where you lose the bridge is that the human relevance, right? So the 3D models that we are creating, these are not animal cells that we use. We use human cells. They could be primary cells or transformed cell lines, but mostly we use human primary cells. And these primary cells can be connected to other cells. As I said, diabetes. Diabetes is a multi-organ functional disease. You need intestinal cells, you need pancreatic cells, you need liver cells, you need muscle cells, you need adipose cells. All this together work in the diabetes disease. Even one organ is dysfunctional, you'll get diabetes. So it's not just the insulin. Production deficiency is not the problem. It's also insulin resistance that comes with other problems of adipose and liver and muscle, etc. So we created a model for that with 5 different cell types: adipose cell types, muscle cell types, a liver cell type, pancreatic cell types, and intestinal cell types. So that will cover all the organs that work with each other in a disease. So that is where we are trying to decode the human biology and in vitro 3D.
Right. So, sir, just out of curiosity, I would like to know how has the industry responded to this 3D bioprinted model? Are they willing to replace the animal model, or they are taking it as an add-on step after the animal model and before it goes for the clinical trial?
I want to call it industry, but the people work for regulations, right? If the regulatory agencies request them, that's what they want to, because at the end of the day, regulatory agencies have their own positions. They have their own guidance documents, regulations, and you have to follow them. So suddenly you say that I'm doing in vitro cells and I'll take the molecule to the clinic, they may not accept, because they'll say, okay, we are regulatory agencies, prove to me that you don't need animals to go to the clinic, right? And this is always a problem. That, I mean, we need to show them what is the value of these models. And in fact, world around, people are trying to develop these models. Some people are focusing on hepatotoxicity models. Some people are focusing on cardiotoxicity models. Some people are focusing on efficacy models. So we have different models that we are creating at different parts of the world. We've chosen efficacy models in our company, and try, because efficacy is the one that the drugs fail in phase 3 trials. You don't want to go to the phase 3 trial and fail in phase 3, which is already spent hundreds of millions of dollars to get there. So in which case, what we're looking at is how these 3D models function like human organs, but yet, and also connect with each other, communicate with each other. As I said, communication is very important. Pancreas produces the insulin, right? Insulin is not produced by the liver. Liver uses the insulin, or muscle cell uses the insulin and takes the glucose. So you need both cell type, right? You cannot just say, hey, I've done in muscle cells that I know how to cure diabetes, but you need to have an integrated model. And these integrated models will help in the knowledge, and then we can design the clinical trials better.
Right. So if you could throw some light on how exactly these 3D printed biomodels are created in your lab.
Yeah, in our lab, what we've done is 3D. You see, you know what? If I take a drug orally, right, it goes to the intestine. From the intestine, through the portal circulation, it goes to the liver. And from the liver, it may be excreted via bile back into the intestine, or it may be metabolized in the liver, and the metabolites go into the rest of the organs. So the drug that goes to the actual tissue that it has to work may be fraction of how much the drug you give. So you need most of the drug to go to the target tissue, right? How do you manage to do it? And that is where our models are going to help to see. We have intestinal layer, we have a liver layer, and we have cancer cell layer, where we put some cancer cells, breast cancer or thoracic cancer, whatever the cancer cells we take from the patients and use them. And this is an integrated model. We know what happens to a drug in the body and how much drug can go into the target tissue. So we can model it. And once we get that data, what we can do is we can create a physiological based pharmacokinetic model. And we use that pharmacokinetic model to predict this much drug will go to the target tissue if you give this much of dose. Is that dose is going to be toxic or non-toxic? These are the things we look at very rationally and try to decide what doses we need to go with.
Right. So now that we have understood a bit about the science part of this, and like you said, business and science, both a person has to learn to run a company, and you have done it very well in last 38 years. If you could throw some light on how exactly the business model runs for Reagene and how exactly you are targeting your clients, if you could throw some light on that.
Right. So this is not a new concept, 3D models. I mean, I said I worked in J&J from 2001 to 2007, and I created a model there. It was the first prototype model there. But the question comes is that, how do you ensure the regulators will accept it? Right? The regulators not accept it, it will not work. So, for example, if I am running the animal business, okay, I want to do animal toxicology that is already accepted by FDA, and I'll create my business. There's the no-brainer, right? If people will come and give me business, because they all want to take their drugs into the clinic, the only accepted model is animal model. But thinking differently here in the 3D model, the idea that here, oh yeah, but I go into the clinic and I'm failing. We need a different model. So when regulators in the last 5 years to 7 years are saying we are ready to accept these models if we have some relevant models. It's not just failing in animal models. Some drugs go into the clinic and fail in the clinic. So I need to actually, they say that you can't run this clinical trial anymore, stop it. FDA will say that. I need to go back to them and say, hey, we have created a 3D model, and 3D model shows that this is relevant. It doesn't have any toxic effects on the humans. So it's okay. It is different from animals, but it is the right way for the humans. So if I can convince them, they'll remove the clinical hold, and I can go back and do the continue in my clinical trial.
Right. So basically, you are helping big pharma companies and companies who are into drug development to overcome this regulatory block using your animal model. That's really smart.
Another advantages I want to point out here.
Okay.
If you want to run a rat and dog toxicology study before you go to the clinic, you need 2 to 3 kilograms of the drug itself, synthesized, manufactured. That's one thing. And the rat and dog 6-month toxicology studies have to be done in GLP regulations. That will take about almost a year to complete both of them. So you're talking about spending about $5 million plus maybe up close to $10 million to do all these things. Whereas my 3D models I can do with a 10 milligram material. I get a better answer than animal model. So, and I can cut the time and cost better than animal models.
And of course, it stops animal cruelty. That also is something I completely agree to.
That is obviously the no-brainer that we have. Animal cruelty will also stop.
Yes.
So
it's a.
But FDA might accept it unless we show these models are better than animal models.
Right. So it's really insightful, and I really feel that vibe that, wow, what a work we are doing at Reagene, and we are not just going to remove this animal cruelty, but at the same time stepping up the game in clinical research and clinical trials. Wow, that's a real thoughtful thing which you and your team has done at Reagene. So what's the vision of Reagene, and how, sir, you are planning to step up the game now that the company has been established and running for a while?
So I'll tell you some gaps in running a company in India versus.
Yeah, that's something.
Those are the challenges that we need to overcome in India, all right? They're like, if I am using human primary cells, right? There are no biobanking in India for primary cells. For example, if I want to do a human hepatocyte, liver parenchymal cells, even today I have to import them from West. And they come in a liquid nitrogen ampoules, we need to cryopreserve them and keep them. Same thing with skin cells. Skin cells are produced by L'Oreal in France, yeah, and then Matec in US. But by the time those cells come to my table, they're almost
due date is over.
yeah, but you can't use them anymore.
Expired.
So what happens is that my window of using them it diminishes. I need to do next 24 hours. I need to finish the experiment.
True. That
So you need to hurry up and you make a lot of mistakes in this one. So the problem is the major gap is that we don't have the reagents that are required to really do the 3D bioprinting that happens in the Western world. If I order a cryopreserved parasite, tomorrow morning it will be on my table in the US. Whereas here it will take about maybe 3 weeks to 4 weeks. But I need to keep the biobanking here and we don't have the biobanks. So we need to have a distributor create a biobank. Nobody wants to do it unless they have enough business to do it. So it's all business centric, right? It's not just science centric anymore. It's also business centric. So basically that's one of the major gaps that we don't. We don't have the highly sophisticated analytical instrumentation. I keep saying I go to all the incubators here, CCAMP, BBC, and many others, even Hyderabad, Biotech. I tell them, why don't you guys buy a mass spec, which is my bread and butter because I've used mass specs in the last 25 years to characterize both the drug and its metabolites. However, I'm not going to use old-fashioned HPLCs anymore. I need a mass spec. LCMS is what I need. A high-resolution mass spectrometry I require, and nobody's buying me. So if I go to these incubation centers, there's nothing they can offer me to really do the things I want. So this is one of the second gap that I see happening. Third is there are no trained personnel. I may be well trained, but I'm not going to keep doing the work in the lab.
true.
I need to go run the business and bring the business to the table.
Correct.
So I need to train people first for 2-3 years. And it's not that I don't want them to leave, but once you train them for 2 to 3 years, somebody will poach them away.
Yeah.
So I become a training ground, not really advancing my business. So these are some of the problems I see that we need to go beyond. And our salaries are also very low compared to Western world. And all the students who work with me, and they say, sir, tell me how I can go to US or Europe and get a PhD like you did. And I can say that, I'm guilty of the same thing, right? I left India to do PhD. So we're not able to create enough job opportunities in India for the people who have graduated. It's a major problem. I get in my LinkedIn almost 7 or 8 requests, sir, do you have a job? Every week I get like that. What do? I pass on to other people who may give them the job because I don't have that kind of money to keep hiring hundreds of people in my lab. So that's one thing we need to create is the job creation in India for MS, MTech, Biotech people who are graduating out of this and they don't want to look at West to go after pursuing their
Higher
ambitions in life.
Right. So, sir, now that you brought the education side of it, and the majority audience of Biotecnika is researchers, PhD scholars, students who want to become scientists just like you in the future. So, what would be your message to these young chaps who want to enter the industry, do some amazing work just like you, or maybe take up your work and take it forward in the coming years? What will be your message, what they should be doing to achieve their goal?
I've been giving talks. I've been giving talks like Rien, and many other places. And the one thing I tell that, first, no offense to anyone here, that faculty must be trained to the state of the art. And the government should give them the grants to be able to train their own students. Not at PhD level, at MS level. If I compare my MS in biochemistry here in Andhra University, it's a bookish knowledge.
Right.
That's not a practical knowledge. Right?
Correct.
that really didn't apply when I go into the lab and to do research. Practical knowledge is totally different from bookish knowledge.
Sure.
So when you do actually practically in the lab, you'll understand what problems you face and how to solve those problems. It will also improve your thinking ability to how to solve the problems, right? So when you're reading books and you pass the exams, you don't know how to troubleshoot anything that you want. So these universities, at least MS level or MTech level, whatever, they should put at least 6 months industry training outside there. UGC has their own regulations how this should be done. So some people tell me I'm on the board of studies for a couple of universities. When I present to them, this is what you should do, they'll say, sir, UGC doesn't allow us to do it. They don't give us money to do it. So we have to follow UGC regulations. So there's a gap between the government and the institutions, how they work together. So you all need to come together and say how good to tell government, hey boss, today is 2024, not 1990.
Right.
We need to change.
I completely echo that, what you just said. We need to change a lot in the education system to make sure we don't have misfits, but people who can actually take the industry forward. We need future leaders than people with bookish knowledge. Completely agree with you, sir, there. So if you look at a student who is coming out of this education system, and of course they will face all these challenges, but what they should be doing right to become a scientist like you, accomplished scientist like you.
So do I call myself a scientist anymore? Maybe not.
Okay.
Okay. So, people still introduce me as a scientist, not as an entrepreneur. I go to a lot of people and they say, hey, he's a scientist. Because that is the thing that they know me of as a scientist. But transitioning from science to business is a different...
It's a difficult.
I'll not touch it right now. But as a scientist, I would say the major thing is that you need to have patience.
True.
I remember where you could also ask other people who did PhDs. It's like 24/7 working. There's no break. Because you need to finish your PhD and you need to make your examiners happy and your advisory board happy and your most importantly your professor happy. So PhD is not a job. You really cannot say, hey, I'll go and do 9 to 5 job in my PhD. That's not going to be the attitude you need for a PhD program. You need to say, hey, I need to spend extra 3 hours to finish this program. You need to have that kind of a patience, determined mind. Nothing is easy in life. Same thing is with PhD. It comes with a cost, and by the time once you finish your PhD, it's not the PhD that is important. I remember my PhD advisor when I was leaving his lab, he said to me, Manny, you got a PhD for thinking, not for just doing some hands-on work in the lab.
Right.
It doesn't mean that I shouldn't do hands-on work. I have to do hands-on work to learn, to get the PhD. But in PhD, they give you PhD for thinking, not just giving hands-on work, but doing hands-on work in the lab. So this is one thing that all PhD students should know, and they should not expect their PhD advisor to solve all their problems and troubleshoot all their problems in the lab.
Very true. Completely agree there. So I think I have asked you enough questions. I hope I didn't miss out on some of them. So to our audience, if I missed out any question, please put that down in the comment section and we'll be sure to ask Vangala sir next time when we meet. So in the meanwhile, sir, it was really fun interacting with you today. Last question, which part of life you enjoyed the most, the scientist life or the business life which you're having now?
Science, I would love to do it myself in the lab, but as you're growing older and all, you won't have the same ability to work round the clock. You need to wear other hats, and especially the training part of it, transfer the knowledge to the next generation. And today we are talking about also how to run a business, not just science. And how do you adapt from science to Today, a lot of young people who are just graduating from with MS and PhDs want to start a company. They don't have enough experience really to take it to the entrepreneurial level. They're learning from the PhD itself. So I used to say, hey, my best part was postdoctoral experience. I got a PhD, but I was not really, it was not really enough to become an independent scientist or independent business. So my postdoctoral experience at Colorado and at Berkeley actually helped me to really know, oh, okay, now I can do anything if I touch it very well. And that kind of confidence you'll get only after postdoc.
Postdoc. All right.
That's my personal experience.
Okay, great. Thank you so much for coming on the show, sir, today. And to all our audience, I hope you learned a lot from Subramanyam Vangala, sir. And to all those who are watching it for the first time, we are going to come up with new episodes every week. This was the first episode where we covered the legend of biotech, Dr. Subramanyam Vangala, sir. And sir, thank you so much for joining us this afternoon for all these insights. We will meet you again very soon in one of the iBiome meetings. And I keep loving those interactions which post on WhatsApp group.
It's not it is not to show off, but it is to very understand.
Right. I learn a lot personally from that. So thank you so much for that as well. And have a great day ahead. Thank you. Bye-bye.